Well, hello and welcome to Coffee Chats with Experts. I'm your host, Laura Moriarty, and today we have the exciting topic of vaccine development and manufacturing. Good morning, everyone.
Good morning.
First up,
Good morning.
Good morning. Just checking you're awake. So first up, let me introduce Candice. Candice is joining us from California. She is a global marketing manager for the immunoassays business here at the Life Sciences Group of Bio-Rad. She started at Bio-Rad as part of R&D. So she was developing assays for our Bio-Plex platform. And then she transitioned most recently into marketing. And throughout the years, she has helped to support hundreds of customers in their work with multiplexed immunoassays. So welcome, Candice.
Thank you, Laura.
All right, next up we have May. May is a staff scientist down in the digital biology group, which is based in the East Bay here in California. And May began her career at Bio-Rad doing qPCR assays and development, and then transitioned over to the droplet digital PCR technology. And she has over a decade experience as well in microfluidic systems engineering, multicolor flow, and 20 years experience in protein quantitation and cell-based assays. And she's currently leading the effort at the Digital Biology Group to develop additional droplet digital PCR biopharma QC kits. So welcome, May.
Thanks, Laura.
And next up, we have Adrian. Hey, Adrian.
Hey, Laura, how's it going?
It's going good. It's going good. Thank you. So Adrian is an FAS manager. So that's a field application scientist manager. And before he was promoted to that senior position, he was a field application scientist himself in the field, helping lots of customers. And previously, before working at Bio-Rad, he has worked for some of the largest vaccine manufacturers in the world. And he has experience in process development for multi-subunit vaccines, as well as primary R&D for adenovirus vector vaccine constructs. And that's hard to say early in the morning. So since then, he has worked at Bio-Rad, and he's been helping people solve lots of complex problems with lots of different applications in the field. So welcome, Adrian.
Thank you. Looking forward to it, Laura.
Awesome. And last but not least is Bill. Hey, Bill.
Good morning, Laura. How are you today?
Good, doing good. So Phil's based over there on the East Coast, and he was complaining about a foggy morning this morning earlier, saying that it was like a pea souper. So I really appreciated that that kind of British historical note there. Thank you.
No problem.
Not that I was old enough to remember the pea soupers. All right, so Bill has been doing process development for over 23 years now, and he has been helping customers all in the field with Bio-Rad for the last 5 years, traveling around the US and providing support in process purification, chromatography applications. All right, welcome, Bill.
Thanks. Look forward to the chat.
Thank you. All right. So before we get started, as always, I just want to let everybody know that we love to get your questions. So please, please enter those into the Q&A box, and we will get them in front of our experts and get you some answers to your concerns. And not to worry, if you're listening and watching on demand, you can still go ahead and enter your questions, and our esteemed panelists will get back in touch with you through the email address that you use to register for the event. All right, I think that is all of the housekeeping items out of the way, so we can start to get ready. Is everybody ready?
Let's go.
Yes, let's go.
Let's do it. All right. So as always, I like to kick us off with a couple of my own questions. So here is the first one. And this question, I guess, could probably go on for the whole of the coffee chat. So let's try and keep it short and sweet so that we can have some space for other people's questions. But first up, what is the most challenging aspect of vaccine development? And who would like to kick us off? Adrian.
Sure. So, I think it really would depend on who you ask on these questions. Vaccines are biologics in the most part. They're incredibly complex to manufacture and design. even down to logistics and shipping, temperature control. So it's a very complex product. Low tolerance for side effects from the customers who want to take it. From my perspective, I used to work in more of the cell line development, kind of closer to upstream manufacturing, where you're actually creating the product before moving it downstream. These molecules are tough to design, they're tough to manufacture. And really the part I had a hard time with was you need to make a lot of it. You're making these out of cell lines, you're getting as much expression as you can without crashing them. They need to be typically conformationally locked or at least full-scale protein. So it's very tough at that part when you're making something at scale to do that.
Fantastic. Bill?
Hey, since I work in the downstream realm, I guess I'll chime in after Adrian. One of the big things with downstream is, a lot of vaccine work isn't necessarily a platform. I mean, you have the traditional egg-based vaccines that, are tried and true, but, this newer technology, you've got mRNA, you've got AAVs and other types of vaccines out there that are now requiring different technologies. So downstream wise, we get a lot of questions regarding, separation of empty versus full and things of that nature. And even as Adrian just mentioned, cold chain, I was reading an article earlier this week where they said upwards of 3 billion people right now would not have access to cold chain vaccines. So, in this era of COVID, I mean, that's a significant number of people that would not be able to get any of the current vaccines under development.
Yeah, you raised some really good issues there. May?
Yeah, I agree with you guys completely. I think the challenge with under the COVID current situation is that the sheer scale of vaccine development. I mean, this is unprecedented. We have 44 clinical trials in phase 2-3, right? And then we have 154 preclinical candidates. And and plus we also have the existing vaccine that we have to make, right? So, so I think that just getting everything manufactured and transferred into a regulated environment to make sure it's safe and efficacious, that is a huge challenge. But I mean, this is unrelated to science right now, but we're seeing in the clinical trials, we're seeing countries coming together. Usually we're used to like the United States, somewhere in Europe, and definitely that's foreigners right now for mRNA. But we're seeing places like Kazakhstan and Cuba, and that coming from China. I think it's just so encouraging that we're all coming together despite, everything else is happening. And that's just really exciting. Everything's going, forget about politics, forget about history. We have an emergency, we're coming together. So I think that is really heartening, But anyway,
yeah.
So.
No, that's it feels like a warm hug to hear you say that. It's absolutely true with everything that's going on. That science is really playing a key part, and we're coming together. I love it. That's a great way to start the coffee chat this morning. Thanks, May. All right, let's move us on to a little bit more of a technical discussion here. So what are the key technologies that assist in vaccine development? And I know that everyone can kind of speak to their experiences on this one. Who would like to give us a shot?
Okay.
All right. Okay, May. Okay.
Yeah, that's a really good question, Laura. It's really broad, but it really starts us thinking, right? And I said, it's just the scale of everything. We have new technologies coming in because we have to really innovate and push things forward. Things like droplet digital PCR, which can fit into multiple levels of the traditional manufacturing of viral-based vaccines. We also have new flavors of existing technologies like tried and true, ELISAs, protein quant Western blotting, and process chromatography. I mean, this is bread and butter, and that is seeing resurgence in innovation as well. So, and also multiplexing of, say, for example, immunoassays. I mean, that is really pushing it forward. And Candace here, my colleague, is definitely the expert on that. And yeah, Candace.
Thanks, May. Nice lead in. All right. Well, I'll go ahead and take it from there. So as May had mentioned, there are a lot of traditional technologies. Multiplex immunoassays is really very similar to an ELISA 96 well plate format. You're able to detect different proteins in the same well due to like different fluorescent signatures for each bead. And so that's what allows you to do that detection. And that's actually used all through vaccine development from, when you're looking at animal models and trying to understand the immune response to your specific vaccine by either looking at, cytokine signatures based on, formulations of adjuvants, looking at antibody titers, both in animal models all the way through to humans. And because of the fact that you can use such a small amount of sample, it's really ideal for those animal studies early on in vaccine development. And then because the format of the immunoassay is so flexible, you can basically develop anything that you would, via ELISA, so competitive assays, sandwich assays, direct and indirect. So it's really very flexible, and you can use it to basically quantitate many different proteins and use it for many samples. So there's a level in which it cuts down your work when you want to test, maybe multiple types of samples longitudinally over time to see what the antibody titer may be after you first initially dose someone with a vaccine, and then you do something like a booster shot later. So it's really quite useful, again, through the continuum of vaccine development.
Adrian.
Yeah, so I'll second this. So I worked on the cell line development side, and initial clone screening was one of my main jobs at a previous company. And we were always limited on how many clones we could pick by the assay side of it. We weren't using as many multiplex at that time. So if we were looking at multiple proteins, especially protein conformation, those would each be a separate assay. So anytime we could multiplex it and increase the throughput that we could do, increased our clones, which is kind of like shots on goals. The clones, I was involved in cell line derived clones, but the more shots on goal you took, the better chance you got of a high quality clone that could produce a lot of protein that was going to meet all of your quality specs that you had picked out front. So it was really important. If we could multiplex something, I mean, that always won the day. That really sped our development time up.
All right, thank you, everybody. So I'm going to hand over to the audience now for some questions. So let's take a look. All right, how about this one? Okay, so recent improvements or developments in HA, HS, and MMC resins. And I'm guessing resins, so I'm guessing this one's for May. No, Bill, go ahead.
As far as recent developments, especially in the mixed mode space, our Nuvia C prime and our Nuvia A prime have shown the potential to actually be able to separate empty versus full capsids. We actually did an adenovirus purification, and when you actually look at the chromatographic profile, you can see there's actually a double peak during the elution. Now, that initial study was just to capture the adenovirus. It wasn't necessarily looking at empty versus full, but it definitely appears to be able to show some separation. So that would definitely be a key aspect to developing vaccines, especially, these AAVs, because you want to make sure empty versus full, you're able to get that separation. And additionally, we have Nuvia HBQ, which is a small 50 micron anion exchanger. However, it was designed with very large pores, 5,000 angstrom. Most resins are about 1,000 angstrom pore size. So you're kind of limited by to surface binding in a lot of the other traditional resins. So Nuvia HBQ would allow a large molecule to actually get inside the bead itself and increase your binding capacity.
Hey, Bill, I have a question. So for our resins, I know CHT has been around for a long time, and I used to use it to separate double-stranded versus single-stranded DNA. I was wondering, is there a difference in terms of resins to separate RNA versus single-stranded DNA, or how is that different?
A lot of it depends on the structure of the molecule. So, I mean, CHT is unique because it actually does bind up phospholipids very strongly. So I'm not sure, depending on the size of the molecule, you might be able to affect separation with the phosphate gradient. But, other resins, they're going to bind the DNA just as well, but you're going to separate it in a separate manner comparatively. We do actually have a lot of vaccine usage for CHT. It's used by a lot of our customers.
Yeah. Great, thanks.
Brilliant. All right. There's nothing like a question from the audience and then a question from the panel. It's like bam, bam. Well done, Bill. Extra coffee for you. All right, next up. All right, so you mentioned droplet digital PCR is used in vaccine development. What about QC? Okay, so can droplet digital technology work further downstream? I guess this one's for May.
No, Adrian. Okay. I'm sure, yeah. So droplet digital PCR for this application is actually kind of a new kid on the block for now. And in vaccine development, I'm not too familiar with the new platforms for mRNA and peptides, but a majority of the candidates are cell-based produced viral vaccines. And for that, the process is pretty much, starts with cell line development, transfections. And then it leads into, separation, chromatography, QC. So for droplet digital PCR, there's applications at the front end for cell line development in the R&D part of it. So there is copy number variation, transgene expression. This is when you're making the cell lines. And then when you go a little bit later on, you can have like functional assays. That's in combination with ELISAs and plaque assays. And even later on, you can check for host residual DNA with ddPCR because it is absolutely quantitative. It gives you a accurate and precise measurement at low quantities. And at the end, when you have, residual, I guess, contaminant testing, droplet digital PCR is used for mycoplasma. So that's just a few. I'm sure I probably missed some, but that's just what comes to mind right now. Yeah.
Brilliant. All right. Okay. Let's pick this one. Why are multiplexed assays used for development? I think you kind of touched upon this a little bit earlier. But one of the benefits.
I can add maybe a couple of other things. So, there are some populations where vaccines don't work as well, like maybe young children and elderly, due to the way that their immune systems are either underdeveloped for young children or maybe going through immune senescence for older people. And so formulation of the adjuvant can be something that people study to basically get the maximum immunogenicity in a vaccine for a specific population. And so multiplex immunoassays in terms of some of our cytokine panels have been used, some of the mouse cytokine panels have been used to look at some of the T cell responses that would allow researchers to study whether or not the vaccine will have that immunogenicity required, because the adjuvant's actually stimulating, the innate immune system in order to support a stronger adaptive immune response and get that higher titer for the different antibodies that you develop against that pathogen. And so it's one of those things where if you're looking at a lot of different formulations and trying to look at different models, that it's really convenient and a nice, high throughput way to evaluate that type of scenario. So early on, I guess, in animal models and when you're developing the vaccines, it's very useful. And then I think I kind of touched on, the continuum. So I'll go ahead and just leave it there.
All right, brilliant. Anybody want to add onto that one for multiplex?
Yeah, I do. So
okay.
I think the bead-based Luminex platform is just really pretty amazing in terms of throughput. Because if you think about traditional flow cytometry, we're talking about maybe like 10, 12, markers. But with the throughput, With the bees technology, I think you're up to what, like 40? Candace, like quite a few, right?
No, you could actually, there's 100 bead region map, but most of the protein assays, due to, the complex interactions with, all the antibodies in there, it's usually less than 50 for the multiplex panels. And those are like large screening panels.
Yeah, so that's amazing in terms of like comparison to ELISAs, where you're only doing like one at a time. So when you look
That's
at
true.
the whole, cytokine profile, that gives you a much more accurate and comprehensive picture of what's happening. So I think that's
actually a great point. Yeah, so people do look at try and look at those cytokine signatures as predictive biomarkers for developing, different formulations for vaccines. And if they can find something that's indicative of a response that they want to elicit, then that's very helpful for the upfront development.
Yeah, exactly. Okay.
Brilliant.
Thanks.
Cool. Thank you, ladies. All right. What to pick next? Oh, sorry, Bill. Go ahead. Sorry.
I just had a follow-up question for Candace. I attended the World Vaccine Congress a couple weeks back, and there was a lot of data shown by multiple companies with their B cell, T cell responses. And one of the questions always came up is, well, how do you compare one company's results versus another to kind of tell whether the vaccines are actually working similarly? So do you have any thoughts on that?
So when you say the companies, you mean like the vaccine manufacturers? How do you compare their results?
Yeah, because I know that's one of the big things where I guess with Operation Warp Speed was that they were supposed to come up with a centralized system for basically determining their efficacy of their vaccines so that they could actually compare like.
Yeah, to determine efficacy, you want to make sure that you're getting or you're eliciting that immune response that you want. And part of that is like looking at, antibody titers and how effective that vaccine is in terms of eliciting that more permanent response where you basically will have a longer lasting, level of antibodies, and that the next exposure would allow you to curb that infection so that there aren't so many symptoms, associated with that next exposure to the pathogen. And so I guess in terms of looking at looking between different vaccine manufacturers, as long as they're presenting similar data sets where they're showing, that there is an immune response based on both maybe, cytokines as well as antibody levels, and that they are able to show that it's a lasting effect. And so one of the things about antibodies is that not all antibodies are equal. Some of them are, less have lower affinity. And so you really want to make sure to get that response where there are higher affinity antibodies, and then there are also different kinds of antibodies. So neutralizing antibodies tend to prevent an infection where they're really blocking the entry of that pathogen from, infecting the cell. And so I think that as long as you, as manufacturers are presenting similar data sets, it's okay to, compare those and move forward. They don't necessarily have to use the same technologies necessarily, but they should be able to point to similar studies. I guess, I don't know if you guys want to add anything to that.
Yeah, Adrian.
The other side of this too is that while there are some assays that are kind of well established, oftentimes what a manufacturer is doing is showing to the FDA or a regulatory body that our assay that detects X relates to this outcome. And so since, we've talked about the different vaccines, we've kind of touched on them. There's egg-based, there's cell-based, there's RNA-based, there's viral-based. You could generate a different vaccine using each one of these 4 technologies, and you're going to get a different output of what a human response is going to be to that. So oftentimes it's not, am I detecting a cytokine at the same level in one kit or another? You choose the kit you're going to have, you do your test, and you say, okay, when I see this output, it results in this efficacy or this result downstream. And so a lot of it is done up front, where you are kind of defining your own criteria of what is successful and what is kind of, they call this quality by design. So as you reach each step, what are you looking for? What are your criteria? Once you've set it and you move on with those studies, how are you hitting that over and over? So it's oftentimes not about how one kit compares to another. It's how does your science continue to work on your established kit that you've chosen, because you're not going to switch midstream from one manufacturer to another. You're going to have determined that up front and then pegged your downstream results to that kit's readout. So, that could be done at the R&D stage, but oftentimes as they get into trials, whatever they've decided is their criteria is how they move ahead.
Yeah. Brilliant. And is that one of the reasons why people like a technology that can be used from, early discovery further into clinical, so they don't have to switch? Yeah. Yeah,
totally. Yeah. That way it's consistent throughout. They're not having to switch criteria.
Got it. Brilliant. We don't want to add any more variation than you get from biology already, right? Okay, let me pick another question. And don't forget, enter your questions into the Q&A box if you have any follow-ups from what we've been discussing. So here we go. Here's another one. So how do you recommend using, or what do you recommend for using for fast and semi-quality purifications for analytical assays for CLD and upstream development? That's a long one. Oh, Adrian, go for it.
So this is pretty common. You kind of alluded to this a little earlier on assays, where somebody likes to pick something at the beginning that they're going to use throughout. So manufacturers oftentimes kind of have an idea of what resins they're going to use in their purification process, or their best guess. Most of these are platform-based technologies. So they like to use ion exchange, CAHT. What they're really looking to do is purify a protein quickly, maybe 70 to 80% pure in one, maybe two steps. So sometimes they use a size exclusion step at the end for analytical reasons. It does a swap over on buffer just naturally if you want to move it into some type of predetermined final formulation buffer. And it also allows you to ready that sample quickly. So usually 1 to 2 steps, ion exchange, mixed mode resin like a CHT. So one of those two, and then maybe size exclusion, and you're done. It's not nearly what they'll do at the final manufacturing stage, but it's quick, and that's why they do it that way.
Yeah, that makes sense. You don't want to invest too much at that point in making it perfect. Bill?
I will add, though, there are, some smaller companies that once they develop something early, they will actually stick with that. We ran into an issue where we had a company got to phase 3 production, and they were using our 20 micron CHT, and they had massive pressure issues. And we're like, it's not really designed to be used at scale. Fortunately, we were able to transition to the 40 micron, and it worked fine. But, there are people that when you get a good process early, they're loath to change it. So sometimes you have to talk them into something that's actually scalable.
Got it. And it can be hard to convince people, right?
Oh yeah.
Oh dear. It's for your own good. All right. Oh, here's a good one. All right. Are there Bio-Rad products? Sorry, let me push it to the audience here. Are there Bio-Rad products to aid in cell line development? Okay. Somebody wants a hand from us.
No, I'll go ahead and
Candace,
take that. I mean, this is probably Adrian's domain, but I'll add a little bit in terms of immunoassays and how they are, the multiplex immunoassays and how they've helped. So he alluded to this a little bit earlier, but a really specific example that I read recently in an article was GSK developed a single-step cloning strategy where they basically were using, traditional ways to go ahead and get one single cell into a well, and then they used an instrument from Solentum, I think, that imaged it to make sure that there was a single cell in there. And then once they were able to have that cell express the different subunits of the vaccine that they were trying to develop in CHO cells, then they used a multiplex immunoassay that they developed using raw beads. So raw beads are available for people to develop their own assays on. So they were able to develop a sandwich assay that was sensitive enough to go ahead and detect these different protein subunits that they were trying to use in their vaccines at a very small, I guess, or actually at a very low level. And so what this allowed them to do is basically cut out the clones that weren't performing the way that they want. So as Adrian had alluded to earlier, this is like a huge investment in time when you have to go ahead and passage all of these clones. And so in that specific instance, they were able to reduce down from something like 40,000 to like 40 to 80 clones that they identified as really ideal to move forward with. And then when you look at comparable, traditional ways to do this, it would be something like 300 clones. So even going from 300 down to 40 is like a huge time savings. And so it really allowed them to screen out the clones that weren't performing well using both this imaging technology as well as multiplex immunoassays. So I thought that that was a really interesting way to apply custom immunoassays to early cell line development.
That's pretty cool. May.
Yeah, to follow up on that, so for droplet digital PCR, I think it's seeing a lot of adoption in the cell line development pipeline. So for one, the copy number variation and transgene expression is very important to establish that cell line in the beginning. And so we have existing copy number variation validated assays and design engine that can help with that. So it depends on what you're looking at. If it's a custom gene, then you can design your own. And there's validated reference assays to normalize against that. And also for transgene expression, like how well your transfection is working, we can use droplet digital PCR to give you a quantitative percentage of your transgene incorporation in a population in one experiment. So that's another thing, is that gives you a number. It gives you a number so that you know it's how much you have. Say one transfection condition gives you 40% incorporation of your gene. So you know that 40%. So you have an idea when you screen, okay, I'm going to expect, water tank. Or that a different transfection method gives you, say, 80. So now right away, you know which one to go with, right? Without having to screen all of these clones. Yeah. And also you can use droplet digital, because if you get a number, you can screen pools of cells instead of having to make, single clones and screen each one. Candace mentioned that you can, kind of skin that cat in a different way. But for droplet digital PCR, you can just have your transfection, you know what your percentage is. You split them up into pools and you screen each pool. And you take a pool that has higher transfection efficiency and then you screen that. So iteratively, you are not, you're saving time, right? So that's one. I don't want to take up all the time, but I think the as fast as we can go, the better, right? As quantitative as we can, because like Adrian said, expressing that protein and purifying it is really an art. And you can't save too much time there. You just have to do it. So if you can save time on the front end or in the back end, that gives you, more space to do the really hard, the really tricky stuff. So that's. Oh, and another application that usually is not really being talked about yet is that once your cell lines are made and you store them, I think we have experience that they can kick stuff out because they don't like having foreign things stuck in there. So to check clonality of your cell line, you can use droplet digital PCR with the same assay where you check for transfection efficiency. So if you have a single clone of your cell line and you store it for however many times and grow it for different generations, and you run that droplet digital PCR assay, you should not see any cluster where you would expect a wild type to be. So if you see it, that means something happened to your cell line. So that would be a QC method, and you don't have to do those complicated single cell imaging. That takes forever, and it's not really quantitative. So that's another, I just want to get that out there.
Yeah, that's really good. Valuable. Yeah, that's a valuable assay for sure. Adrian.
Yeah, I have to say that I'm kind of jealous. I read that paper that Candace is talking about where they use the custom conjugated bees to cut down their clones. So when I used to do that work, that lab there was doing 4 times the amount of cell clones than I was doing at the time. And I remember there were instances where we were waiting on the analytical department to get through all of our assays to let us know which ones to cut down. So we at times had to passage clones just because analytics couldn't get there. And who can blame them? I mean, you're doing multiple ELISAs on 10,000 clones at the time, and it just takes time to get quality data. I can imagine it saved a lot of risks moving to that type of bead-based automated application. So I'm a little jealous and wish we would have had it when I was doing cell line development.
It's so funny. There's so many technologies that have come out these days that I say, oh, I'm jealous. Why didn't I have that when I was in the lab? So I totally get it. All right, let's move us on to another question. I want to make sure we're getting through to answer the questions from the audience here. Okay, I read about platform technologies being used for the COVID vaccine. Why is that? I guess maybe you could start off with what is a platform technology? Bill.
Well, at least from a downstream perspective, the platform technology is basically the same purification scheme that they use for all of their products. What this does, you basically have kind of an outline of your purification process so that when you get a new candidate, you're able to process it quickly through to final product. Might be some minor tweaks with your elution conditions here and there, but for the most part, your materials are so similar that you're not going to have to do a lot of tweaking, so you can actually get out and get into the clinic very quickly.
Now that makes sense. So reduce, reuse, but I guess not recycle. Or maybe recycle. Oh, I saw Adrian and Candace put their hands up. Ooh, ladies first, Candace.
Okay, thanks, Laura. So something I had mentioned earlier about formulation of adjuvants, I did read that during this time that some manufacturers who had already established, high immunogenicity, but a nice safety profile for some adjuvants were going ahead and, offering it to these manufacturers. So these other manufacturers that were developing different vaccines. And so in the spirit of cooperativity, I thought that was quite nice to see that, people or manufacturers are helping one another develop things faster by letting them use a formulation that they already know works well and that is safe.
Adrian, you still want to add? Yeah.
Yeah, so, platform technologies are used throughout biologics manufacturing. So the quality and safety standards that are involved with this category of drug are just immense. And so a platform means it's been used before. So when you think about vaccines, people want to cut down cost. When you think about a COVID vaccine, they want to increase the speed. So something that's already been used, already been through the clinic before, maybe has a product associated with it. You have a lot of data already. So using the existing platform really lowers the threshold of novelness in what you're doing. It increases the chance that you're going to be able to make this product successfully. And it gives you a little bit more information on the safety side because you kind of know everything, to the point that Candace made with adjuvants, it's been used before, so its safety profile. So you're not changing everything in the manufacturing process. I guess you can think of it kind of like the scientific method for manufacturing. Change as few things as you can, and you kind of have a better idea of what's going to happen at the end product.
Brilliant. May.
Okay, I'm going to add to that. I think you guys are absolutely right. So the tried and true platforms, I think, is a little bit slower to get to the clinic. So right now, I think it's really exciting that we're seeing these other platforms like mRNA and protein and DNA plasmid. And it's exciting. The science is exciting, and they're getting to the clinic quicker. But I don't want to say anything negative. I think it's wonderful. But what we're not talking about is delivery. This is why these platforms has not been widely used before, is that naked mRNA and DNA is very unstable, and it's very hard to get them into the cell. Same with protein, peptides. They work great in a petri dish, but you put them in an animal or a person, like they don't get it. So how do you deliver that? And right now it's lipo nanoparticles, and we're not seeing too much talk about that. Right now it's just getting into it. So how do you get that in, and how do you make it? So that needs to be scaled up too, and how are we going to check that? I'm sorry. If I had to bet money, if it's a vaccine fourth place, I'm betting on what's tried in viral-based, manufactured vaccines that we know it is. So what is takes a little bit longer, but we know it's safe. We know where the bottlenecks and pain points are. I'm a little. I'm curious to see how they're going to solve that problem. Are you going to there's cold chain problems, stability is an issue, delivery is an issue, and I want to see how they solve it. We have to solve it, right? We want to use it. So yeah, great.
Yeah, I want them to solve it so we can get back into regular life for sure. Bill,
I'm going to jump back in real quick.
Sure.
Just speaking to Adrian, the Janssen vaccine is on their Ad26, which was approved for Ebola. But even that, you see, they have just had a clinical hold announced. So even one that's already been approved with that platform technology, they have to actually kind of have this trial pause. But also speaking to May, as you said about the transport, there is a company, I can't remember the name. They just started a clinical trial on an oral version of the vaccine. So if that actually does pan out, I mean, if you think about it, that eliminates that 3 billion without access to cold chain that I was mentioning before. And again, traditional vaccines, we're not usually making 7 billion doses. And actually a lot of these are 2. So you're looking at 15 billion doses. Then if you're doing it yearly, it's another 7.5 billion. So, I mean, it's a lot of vaccine that's going to have to be made over a very short span of time. So I don't know how soon till we get back to, the normal that Laura is, actually, we're all basically screaming for.
Agreed. Absolutely agreed. Couldn't have said it better myself. All right, let's go to a fresh question from the audience here. Oh, so this person's looking for help. So how can Bio-Rad help me check the quality of my protein-based vaccine? I would suggest give a call to tech support, but for some deep dive help, but who has something they think might be able to help this person out? Adrian.
So there's a lot of different ways that quality can be looked at, but I'm going to look at it as, since it's protein-based, the actual quality of the protein itself. So a lot of times outside of doing ELISAs, people want to make sure that the protein is being expressed in full length. They might be looking at modifications. If it's not since we're not talking about RNA or DNA-based vaccines, they're going to want to make sure the conformation of the protein, sometimes that's very important. It needs to be conformationally locked. So a lot of people move to multiplexing Western blots. They might use a GS900. So if you're looking at quality as far as instead of what protein do I have, which is a Western blot, it's going to tell you that, or what conformation or what subunits, which a multiplexing fluorescence is going to help you, like the ChemiDoc MP. Sometimes quality means protein contaminants. We've made a protein, we've vialed it. What kind of host cell proteins are we looking at? So the GS900 imager can be used with Kumasi imaging. It's kind of the gold standard for looking at protein contaminants within the QC area. So, depending on what parts of quality, at least on the protein and integrity size, a lot of times it's imaging.
Brilliant. Fantastic. I felt like I needed to do a game of bingo for how many Bio-Rad products you mentioned with that one. So I didn't quite get a line. Okay, let's go. Okay, here we go. All right, and I read this one wrong. What type of, I almost read texting. What type of testing is done to check vaccines are conferring immunity? And I think Candice kind of talked about this earlier, but maybe we can just, Candice.
Yeah, so with regards to multiplex amino acids, I'd mentioned already that we look at titers for antibodies. You could basically coat the bead with whatever specific antigen that vaccine, is relevant for. And you can look at the fact that in that immunized either animal model or human, that they're actually generating the immune response that you expect from that vaccination. And then the other aspect with the flexibility in being able to develop different formats of immunoassays on the bead, you can actually also look at the neutralizing antibody effects as well, where you can have a competitive assay that shows whether or not your vaccine is actually eliciting neutralizing antibodies, which tend to be the ones that are more efficacious because of the fact that they can prevent actual infection from happening. And so in that case, the traditional technologies are PRNT, which is like a cell-based assay that basically puts you in the lab for 3 days, and you need a BSL-3 level lab. And so at that point, this is reducing it down to like less than basically 3 hours, where you could get similar results. And then there's also a pseudovirus test that still takes 2 days, but it does lower your BSL level to 2. So it's one of those things where there are many different ways to check whether or not you're getting the effect that you want. And I think that Adrian and I were chatting about this earlier too, just that you actually have to check it among the population at large later to make sure that when they are challenged again, that they are not going to get sick. So you need the real life data as well as what you get in the lab. So,
Adrian.
Yeah, that's right. That's what we're seeing in the news right now, where they talk about what's the delay or when the date's going to come out. When it comes to coronavirus, they're looking for 2 months of data after somebody's been gotten their final immunization. I looked at the criteria they ask volunteers for. It's very evident they're looking for people in, kind of high-risk jobs where they're going to be exposed to it. They want people to get sick. They want to make sure what they're looking at in place in some countries. We're not doing it here. The UK has started a live, or will be starting a live virus challenge. So they'll immunize people, and then a few weeks later, they'll actually inject them with a purified COVID or SARS-CoV-2 virus, and then see how many people are immune versus the control, and find out how many get sick. So there's a lot of different ways to check the immunity of it, and it's why they do, phase 3 trials. Is it safe, and will it give immunity for whatever period of time that trial is set up for?
Yeah, great points there. May.
Yeah, this is a little bit of a fun question, because we were talking about, like, you had mentioned that you volunteer for trials a long time ago. So what do you think? Like, is this, would you volunteer for this? You don't have to answer, but what do you think? Do you think it's safe right now?
To volunteer
I think
for...
plead the fifth, as they say in America.
Yeah, I mean, it's as safe as getting sick is. I mean, that's at the end of the day, the risk is similar to a live infection when you're looking at a live challenge. So, they've done live challenges in the past. It usually depends on how bad the disease is. It's not a completely uncommon way to do these trials. I was at one company where instead of injecting people, they were going to inject individuals. This was a malaria vaccine. They were going to inject U.S. soldiers who were going down to Africa, someplace where malaria was endemic. And then they were going to see how many of them got sick, just let them work like they normally do. So, this does happen in vaccine trials, and it is a speedy way to do it. But I think in this virus, cost, the benefit may outweigh the cost, but that's an individual decision. No way to know what's going to happen once you get sick.
Yeah, I think the ethics around that are, that would be a whole other coffee chat for sure. All right, let's move us away from that, from those thoughts into this one. And now I wish I'd got to this one earlier, because I'm not exactly sure which product they're talking about or which technology, but this person's asking, does Bio-Rad have GMP-approved software? And I don't know which product it's for, so maybe you can speak to the products that you know.
Sounds like a marketing question. So I I think that many of our instruments that we expect to, be used in industry have software that enable GMP compliance. And so oftentimes at Bio-Rad, it's called Security Edition or, 21 CFR Part 11 software. But really, it's up to that individual lab to make sure they're in compliance, but we're providing them the tools by which they can do that. So I know we have it for things like Image Lab, which are for our imagers. We have it for Bio-Plex Manager, which is for our multiplex immunoassay hardware. We have it for the GS900, which is also actually, I think, Image Lab. I don't know, for NGC, we have Chrome Lab software, that's Security Edition. You might want to weigh in on the genomic stuff.
Yeah, I'm sorry. Yes, we do, because that is a huge effort here. So for the droplet digital PCR QC kits, we are, we are pushing it out with 21 CFR compliance software with GMP environment in mind. So yes, we are definitely doing that. We're serious about doing this.
Fantastic. And I guess that speaks to what Adrian was saying earlier about having a technology that can be used early in development and then all the way through to manufacturing and QC. All right. So I'm going to see if I can get us through these last couple of questions here. So we're going to do kind of rapid fire. So I'm going to pick this one first. Okay. And I think this one, probably May or Adrian for this one. So how can digital PCR assist with viral quantification?
Okay, so as of now, the droplet digital PCR is actually becoming the industry standard for titering. And that's because of the absolute quantification without a standard curve. So qPCR has its advantages, but it requires a standard curve. And usually when you have inaccuracy, it's because that standard curve material is not well characterized, or it degrades, or whatever it is. So if you base your quantification on that, then your numbers will be off. So with droplet digital PCR, you're getting absolute quantification with the precision and accuracy without having to rely on a standard curve. So you take that out of the equation. So this is why the industry is switching over to droplet digital PCR. And on top of that, the droplet digital platform that we have has been around for a while. So that platform is stable and it's ready to go into GMP. So that on top of the technology and the stability and the quality that droplet digital PCR gives you, that is a leg up from the other digital PCR platforms. So this is why it's better.
Got it. Beautiful. All right, here's another one that's kind of broad, but what does Bio-Rad offer for screening? But I don't know screening of what. Maybe Bill, you can talk about it from I
can talk as far as from a resin standpoint.
Yeah, that'd be great.
Yeah. I mean, we have loose resin, but we also have 96 well microtiter plates if you want to do high throughput screening. We have various size robo columns. We have pre-packed columns of most of our resins. So a lot of things that you can just then get without having to pack columns yourself. It's kind of funny, over my years, it seems like packing small scale columns is becoming a lost art form, because you talk to companies nowadays and they don't teach this. So it's like I walk in there and I'm like, really? I'm like, all the columns I packed over the years, and now I can just like grab one off the shelf, and it's like, it's kind of frustrating sometimes.
Oh, I totally get that. And yeah, I guess it saves time, increases reproducibility, but yeah, there's an art and a basic understanding of science, I think, that's lost when people are given, here you go, just put this with this and off you go. But okay. All right. So I'm going to keep us going so we can get through these last couple. So is there any new technology that's making a difference in vaccine development? And I guess it doesn't have to be Bio-Rad, it could be something else. What are you guys hearing out there?
That's another really great question. It's very broad. So the new technologies, like I mentioned before, it's widespread because we have the need to speed up and scale up. So of course, I'm impartial, right? Of course, I just think that droplet digital PCR is kind of like starting a new, it's kind of like starting a new trend because we need quantitative, at least semi-quantitative assays, analytical assays that lets you measure and it gives you a good QC spec. So, so we're seeing that more and more because we need the comparability, reproducibility, and traceability. So we're seeing that across the board. And I think maybe droplet digital PCR is leading a little bit there, but we see that in like the multiplex immunoassays as well, right? That's semi-quantitative. And so we're seeing that, and of course there's the new resins, the new combinations and the ready to use. And then another big thing right now is the single-use production for cell line development, bioprocessing, is that instead of using the same consumable over and risking contamination, you're just buying something off the shelf, use it once and throw it away. So that's been something that's been talked about by chemical engineers for a while, but it's actually seeing a big, it's getting a lot of press right now. So I think that's pretty exciting.
Getting adopted. That's brilliant. All right, let's get us onto this last question here. And I kept this one for the end because I thought it was a nice way to finish it up for everyone. So how do we ensure a vaccine is safe when development is happening so fast? And I think that this is something that's being felt by scientists and non-scientists alike. Who would like to take a stab at this one? Adrian.
All right. So, this is, of course, the question I think that's at the center of everyone's mind. Typical vaccines take about 10 years to develop. So something that can be done in a year is lightning fast. Maybe 5 years would be considered pretty quick, but a year is just amazing. There's an enormous amount of effort that goes into the quality of these vaccines. One of the things I like to talk about with it is, if you have a heart condition, you're going to take a pill with probably some nasty side effects because you already have some type of pathology that it could kill you. With a vaccine, for the most part, you're healthy before you get it, and you want to remain healthy and safe afterwards. It would kill a vaccine's draw if there were side effects that healthy individuals had. So I had read a quote by a CEO of a large manufacturer that said that up to 70% of their production time was dedicated to quality tests and making sure that this happens. They use a process called quality by design, which is really their way from kind of an upper level position of making sure that everything that's going into the vaccine has been defined, that the quality is high, they know what's going to come out of it. I'm actually kind of encouraged by what we're seeing. We've seen 2 large pharmaceutical companies put a pause on their vaccine trials right now. They've been in the news. I've had some people say, well, that's probably bad for their stock. I think it's good. The last thing you want to do is put a vaccine out there that doesn't work. And so seeing companies that are in a race to solve this gigantic problem pause and take a minute, that's encouraging to me. It says they're looking at the bigger picture beyond, let's just put some stuff in someone's arm and make some money. They're really thinking about what impact this is going to have down the road. So that's kind of what I think about what they do to ensure quality. That it's throughout the entire process by defined from the get-go.
I can't hear you, Laura, but hopefully.
Sorry, go ahead, Bill. You've got 30 seconds.
I just wanted to say real quick, people think fast, and they're like, part of it is the governments are investing in these companies, so they're going to make hundreds of millions of doses that some of these will just get thrown out. Companies typically won't make all this stuff, without knowing what's going on. That's why it usually takes so long. It takes 8 to 10 years to get the data, and then another couple of years to generate enough doses to get out into, to vaccinating. So they're going at risk, but it's not entirely at risk. Obviously, governments are giving money to have these doses at the ready if, when they get to the endpoints of their trials, that they're safe. And, you know, the regulatory agencies are reviewing stuff kind of at the same time, which usually doesn't happen as well. So it's not that they're cutting corners, they're just kind of doing multiple steps at the same time.
Brilliant. Okay. I think that that's a really good way to look at it. Thank you, everyone, for your questions in the audience. And thank you to our esteemed panel. I really enjoyed the discussion this morning. And last but not least, it just would like to give a quick plug to some of our other coffee chats with exciting topics such as qPCR for infectious disease, tips and tricks for Western blotting, and tips and tricks for chromatography protein purification. All right, so that is it from us over here at Coffee Chat Central. I would like to, say a big thank you again to our panelists, and I'm going to sign off for now. Take care, everyone, and stay safe.